Just to announce the next talk, as promised, let's say in my introduction, we have now a talk about the thyroid gland. I think everyone has some background on thyroid function, what it does, but to go into some more in details on the microstructure and function of the thyroid gland may relate to feed efficiency in the bovine, and this is a presentation from Canada actually, and the presenter is Yuri Montanhori. Hope that's pronounced correctly. Okay, go.
Thanks, Dr. Servin. It's really a great pleasure to have invited a presentation in the Animal Physiology section. It's really like a shining star in my career, and I really value this opportunity. So I'm coming from Canada, which is just across the pond from here. And I'm coming from Nova Scotia, which is one of the Atlantic provinces in Canada that's very famous for seafood production and also blueberries. Is actually right now is the peak of the blueberry season there, is everybody has their mouth kind of blue. And we also have some cattle there, not that much. And that region was really affected by the mad cow disease about 2003, 2004, which really decreased the herd size. And now we are working on that, trying to rebuild the herd size there. And hopefully that in 10 years from now, we have the same herd size that we have in 2003. So just for you to see how bad a disease, how many bad things a disease can cause to a production system. Despite the difference, the distance that you are from home here, there's an interesting connection between Belfast and Nova Scotia. While here, one day, we built what we consider that to be unsinkable. In Nova Scotia, we have a quite interesting museum about the wreckage, and have a lot of the floating debris on that museum. So for those of you that like history, it's a great museum for us to take a look. I am a big fan of the EAAP meeting. I started following this meeting in 2010 with a good excuse to get to see Greece. And then I went to Slovakia and then Denmark. And what really brings me to Europe is the broad scope of this conference, and the value to the diversity, to the small farming systems that you don't see so much in the other side of the pond. Last year, I managed to bring my whole lab to the EAAP in Poland. It was a great experience for all of us. It was me, my 4 master students, and an undergrad student that worked with us. That we did a lot of presentations in the different areas that are bound together to talk about feed efficiency. So our goal is really to understand how different systems, how different metabolic systems might be related to feed efficiency. And today I'm carrying the torch of our lab here to talk about thyroid and how that 44 grams of 1,000 kilo bowls matter so much to many of the physiological systems that we have been studying. I decided to divide my presentation in 2 main portions. The first one is really to talk some simple examples, more to warm you up and get used to my accent. And then the second one is where comes the heavy stuff, where we talk about some of the more complex examples. Sorry. So we start with the simple examples. I'll be talking about some of the applications that you might be observing for like applications that you can use thyroid to access Oestrous, also to access the presence of or absence of liver abscess, and also what's the use of thyroid hormones when you're doing spot sampling for blood, and how that might relate to feed efficiency. So we start with Oestrous. We did a pretty laborious work where you're blood sampling heifers every time that they show Oestrous. And those heifers are located 130 kilometers from our office, so we had to drive every day 130 kilometers, check the heifer, see who is in Oestrous, get a blood sample, and come back home. And despite the work related to that, it was a quite interesting study. We measured over 40 different blood analytes on those animals. And in this graph here, I have a short list of the analytes that proved to be some relevance to detecting Oestrous. So this analysis was done using logistic regression, and every metabolite that has a certainty higher than 1 was included in here. So you can see that some analytes like ALP, AST, BHPA, and CK, they are just about 1, while when you look to T3, that was pretty high, was like over 3. So really showing that T3 has a big impact in telling if the animal is in estrus or not. And when you put all this together, just by the blood work, we could have a certainty of 92% that the animal is in estrus based on those 5 analytes. So this is interesting for the physiological side. I'm not advertising here that you should be blood sampling animals to tell if they are in estrus or not, but there's some relevance on that to really illustrate that when the animal is in estrus, the animal is in an anabolic state, and when is that in that anabolic state, T3 is a major player on that. And I have some more to talk about estrus detection. This is a presentation that we have on the section 70, that will be on Thursday. This section I'm chairing and also presenting this study here. This is all done in hosting cows. There is no blood involved here. There's quite a few other analytes that are looking at the animal and other parameters to say if the heifers are in estrus or not. So another interesting thing that we learn about thyroid is the connection with liver abscess. And this comes down to my days during vet school, where you have your inspection class, and then the prof take you to the abattoir and show, oh, this is the liver, and then you see people doing some massage in the liver, and then do 2 or 3 cuts and say, oh, this is good. And I never bought that. I always found that could be quite misleading, because you could have some small abscesses here and there that you wouldn't be picking up with that quick massage and 2 or 3 cuts on it. So a little while ago, I had the opportunity to run an experiment on this and learn a bit more about that. And liver abscess can be a pretty big deal. That 20% is the prevalence of a liver abscess in the Canadian herd. And that's an underestimation. It's actually more than that. And so what happens with the liver abscess? You first are condemning an organ with like 4 or 5 kilos that's going to the drain, or getting devalued, becoming soap, whatever it is. And then animals that have the liver abscess, they have a reduction on their performance. So they have effects on body composition, fetal gain, and average daily gain. So that really causes a chronic problem to the production system. And what we did here, we compare animals that had and animals that didn't have liver abscesses through a series of different analytes. So I start from the 3 from the bottom first. You have their glucose, albumin, and cholesterol. And then you see that all those analytes, they are increased in the normal liver and decreased in the sick liver, in the abscessed liver, which make more sense because those are really associated to liver metabolism. So if the liver is sick, then you might have a problem in, let's say, synthesizing albumin. So that really illustrates that there is a metabolic difference on that. And then comes the thyroid hormones. T3 didn't tell us a thing. There was no difference in T3 between animals that has abscess or not. But when you look to T4, there was a difference. Animals that have abscess, they have lower levels of T4. And which also make a lot of sense because we know that liver is a primary organ for metabolizing T4 into T3. So that's quite interesting to see that you may, like the T4 might be part of a package for assessing liver abscess. And how I see this moving on to some application. I think that at some point in slaughterhouses, when you just stick the animal for bleeding, you should have some sort of a blood analysis there, and from there have a better perception of what might be good, what might be wrong about that carcass. So I don't think that that's happening next year, but I see that in the way that you are automating, you're becoming more and more careful with the food chain. This might become a reality at some point. And who knows, you might have a genetic company that also wants to jump on that will make things even easier then. Okay, so now we'll be jumping to what's the main portion of this presentation, that it is the associations of thyroid hormones with feed efficiency. And there are so many ways to define feed efficiency. I'm talking here about residual feed intake. So residual feed intake is a measurement that's like, is a growing measurement that's like getting more and more application in beef cattle production, and is actually part of quite a few breeding programs, which really shows that there is a industry commitment to it, and what makes my job easier as a researcher to do work related to that. So this concept is pretty old. Actually, the first citation is from 1941 in poultry, but we're skipping that. We're going to the first definition in cattle. That's from 1963 from Robert Koch. And what they did here, they just did a simple regression of feed intake on body weight and body weight variation, which is in our case growing animals average daily gain. And with that prediction, they calculate a residual of the observed feed intake minus the predicted value. So an animal that has a negative RFI is more efficient. The animal that has a positive RFI is less efficient, basically. The work from Koch is a good work, it's a pioneer work, but we see some limitations with it. When Koch did the model, he only account for average daily gain without talk about the composition of the gain. And we know that the composition of the gain is a big deal, and you can really be changing animal body composition over generations if you don't take care about the efficiency of fat and protein deposition. So that's why we did some more work on this model, and then we include some indicators of body composition, which are the practical ones for us work with beef cattle that are ultrasound assessments. So we have back fat, we have marbling and rump fat as indicators of fatness, and then we have ribeye area as an indicator of leanness. So with that, we could accommodate some of the variation in body composition, and this has been working very well. So this works very well for growing animals, so steers and bulls, but then we have a complication here. We have the heifers, and those heifers get pregnant. So your model gets a next level of complexity because you have the fetus growing on that heifer that's pregnant. So there's two approaches to it, or two examples. When you see that NRC graph is really the gravimetric relationship with pregnancy, days in pregnancy and fetal weight. So you see that really increases. I think that is pretty obvious that. And the other illustration, that infrared image, is showing the right side of the cow. And then you see that red spot at the bottom there. That's Junior. So Junior has a way higher metabolic rate than his mom. And that really creates that thermal print there. So that really also indicates us that we need to be a bit careful because the fetus has a higher metabolic rate. So accounting for that somehow is really important. We don't have many ways of accounting for this fetus or gestation effect other than fitting days in gestation the model. We could do something like when the calf is born and use the weight of the calf, but you want to do that like early, you only don't want to be waiting for 9 months. Anyway, what you do with the heifers is really to include the gestation, days in gestation in the model to minimize some of the variation due to that. Okay, here, this is an important slide. So if I were to pay attention to one slide, this one, so then you can understand the rest of the presentation. This is a study that we did with commercial heifers. We had access to 107 heifers from 20 different producers. So it was kind of a game to get all those heifers in line to do the work. And those animals we get done in test for 120 days, and every 28 days we did an assessment on them. So this is very like industry-based. That's normally how we test animals in Canada. And because I talk a lot to producers, I'll be using some of the same terminology just to make this simpler. When you do the RFI, the residual feed intake, you have a normal distribution to that of your residuals. And then one way to deal with that data is to dichotomize it, so talk about efficient and inefficient animals, and then you move from there. So that's exactly what I'm doing here. The devil cow are the ones that have a positive RFI, those are bad, those are inefficient animals. And then the angel cow is the one that's efficient, has a negative RFI. So you'll be seeing those two cows throughout my entire presentation now. And also, just to make sure that there is a good understanding of what happens with this working with feed efficiency, I put this slide that to have a lot of performance data here. And what you see here, the first one there we have RFI. So our efficient heifers, they have a negative RFI, so that's dry matter intake a day difference. And then the efficient ones, they have a positive RFI. So basically, when you look to those two groups, there's about a 1.7 kilos of dry matter intake difference a day per animal for feed intake. And of course, that feed to gain is different, with the inefficient ones consuming more to obtain the same gain. And then because we adjust our model for a validated gain, body weight, rib area, and back fat, those traits are not different. So that's quite important because the difference in the animals here is really feed intake, and now adjust for the same productive performance. Okay, so we found some stuff in these heifers. This is one of the, for me, is the most magic pivot that you can get, the 0.06, that makes you think like, why you didn't sample another 20 cows to make that better or make it worse? Just sits on top of the bridge there. But really what we'll be seeing here is a suggestion that the inefficient heifers have a higher level of T3, and that's like measured throughout the test. So every 28 days, like repeated samples. And then we check some other blood analytes on those heifers. Those are part of the complete blood cell count. So we check mean cell hemoglobin and red blood cell count. Mean cell hemoglobin, for those of you that's not quite familiar with this analysis, is a measure of the load of hemoglobin that you have in a red cell. So that's really indicating us that the inefficient animals, they have a heavier load of hemoglobin, so probably a higher demand, a higher workload on those cells for carrying oxygen, while they still have the same number of red blood cells. So there's probably some differences in the way that the red blood cells work when you compare inefficient to efficient animals. And this, when I start seeing this, that again bring me back to my days in the vet school, when you're like there studying, that you see, a cat that has hyperthyroidism is very skinny, doesn't sleep, and kind of destroys our house. And then you have an animal that is hypothyroidism, is fat and very high, very good feed conversion rate. So that kind of makes sense when I start seeing that with our heifers. Of course, this is a pathological condition. It's just an illustration to people really remember what we mean by hyper and hypo, and like increased levels or decreased levels of T3. Okay, and then we did a similar study with feedlot bulls. Our fashion of sampling here was a bit different. We sampled the bulls quite often, over 33 days. So that's like right at the end of the testing period, of a regular testing period. And we observed there was a statistical difference here, that our inefficient bulls, they had higher levels of T3 during that period. So it's really suggesting us that when you're like working in a farm environment, when you're just doing your regular husbandry and blood sampling animals, with a few repeated samples, T3 might be a good indicator of the feed efficiency status of the animal. So that's what I have for the bulls. And one thing that this slide is not on feed efficiency per se, but it's really related to husbandry and related to welfare, which I find quite interesting when I saw these results. So we have a few parameters that we check in the animal to see how is our handling, how is transportation, and how well we are taking care of our animals, pretty much. And we have some parameters that's very classical, like creatine kinase, cortisol, both of them indicating that on farm those parameters have a lower value, but then when you move the animal load and measure that in the slaughterhouse, those values get increased. This is nothing new. People probably know this since the 1950s. Same thing for osmolality. You have the dehydration of the animal with the transportation, and that increases the osmolality. So that's what I have in there. And what's for me is quite interesting here is that when we expose the animals to that stress, there was no difference at all to what you see on farm or slaughter with T3, but there was a difference on T4. T4 was increased when we checked that in slaughter. So that makes me wonder if we can use T4 as a biomarker of welfare and other parameters. And I tell you, over this summer when I was preparing this presentation, I really changed my conception about T4. I think T4 is a very underpriced pro-hormone. People, oh, don't measure T4, T4 is useless, blah, blah. And the more I see T4, the more interesting thing I find about it. So I think that you should look to that pro-hormone a bit more carefully. Okay, so now we'll be moving to the main meal here, where I'll be talking to some of the more challenging studies that we did. So the first one is related to circadian patterns of T3 and T4, and the second one is some relationships with thyroid and other organs and tissue structure. Okay, so I start with a group of heifers. These heifers, I follow them from weaning until they drop in the first calf on the ground. So we had a very long lasting relationship to make this, to get this going. And what we did to those heifers, we checked the circadian blood profile, so hourly blood sampling for 24 hours at 3 stages of their development, when they are around puberty age, when they are like early pregnancy, and then when they are late pregnancy. And of course, for doing that, for bleeding animals every 1 hour, we had to place catheters and then have a long extension so you could be doing the sample without disturbing the animals too much. And this is my first set of results about that. I think that this is a good quality control slide to show you that what we did probably makes sense. Because you see that when the animals are in puberty, so the top line, that's where we have the higher levels of T3, and then early in gestation is intermediary, and then late in gestation, that's the lower levels of T3. So we know that the younger the animal, the higher will be the T3 levels. And also, there's a lot of information that as gestation progresses, the levels of T3 decreases. There are some studies in human suggesting that has to do with increased excretion of iodine through urine. There's not much clear cut on that, but there's some other studies that also suggest that similar type of pattern. Okay, so when you look to T4, it was interesting to observe that at puberty we had the higher levels, which matches the higher levels of T3. That makes sense. But we didn't find difference in T4 over the 2 gestation stages, early or late gestation, just like a similar pattern for those. Okay, so this is like now our clues, our relationships with feed efficiency. And we have our 2 lines here that show that our inefficient heifers, they have higher levels of T3 at puberty stage. That's very important, this stage right now. In comparison to the efficient heifers that have a lower level of T3. So this makes sense, and everything that I'm showing here really matches to what I just said before about the spot sampling. That, okay, you have a higher levels of T3 in an inefficient animal. So was everything going well here, up to here? And then we got here, we got gestation. And when you got gestations, that just blow away that whole story. Because what happens when our heifers got pregnant, that relationship completely shifted from early pregnancy, which is the first 3 months, to late pregnancy at the last 45 days. What we observe here is that our efficient heifers, they start having higher levels of T3 throughout the circadian period on those 2 stages. And that's like, we double check, we think that they're wrong, and then do a lot of work on that, but no, that's what it is. When the heifers were gestating, that relationship with feed efficiency and thyroid hormone completely went in the positive ways, to what we saw before. And then we have some information T4. T4 didn't differ at puberty between efficient and inefficient. It was quite steady on that. But then when you look to T4 at early and late pregnancy, also matches what we found for T3. That's like a higher T4 for the inefficient on both situations. So that's what you have me observe on that. And because we did a lot of work with these heifers, we want to keep them around because they become very tame, so it was very easy to handle them. They are almost like responded by their names. So what we did to them as well, we decided to do calorimetry assessment, so oxygen consumption over 24 hours using head chambers. So that's a system that I was quite involved in the setting up of that system, and works pretty well. It just takes a lot of work to train an animal to do that work well, but once you have the animals trained, it's just perfect and very inexpensive. And okay, what we got here from the calorimetry. So this is a graph that is very easy to draw, but takes so much work to get the data for it. Here we have the curve of T3 and oxygen consumption. So things are making sense. T3 is really an anabolic hormone, and then the curve of T3 is matching very well with the oxygen consumption. So that's great to see that. It's almost like a validation of our system, basically. And then this is what we observe late in pregnancy for oxygen consumption on top there. So oxygen consumption was higher for efficient heifers, so they're consuming more oxygen than the inefficient ones. So they're really capturing more oxygen and utilizing that. And that really matches the curve that we just presented before for T3. So there is some interesting biology going there when you look to the whole animal side. And because research is a lot of time is like gambling, you do something and then you win, and then you want to do the next step of it. So what we did, we also, at the end of the 24 hours of whole body gas sampling, we capture a biopsy of the liver to check oxygen consumption in the liver. So we did that using this amazing machine here that on that time I had under $2,000 budget to get it going. So that's why I use a bucket there for warming up water. So it's quite a bit of cowboy things here, but the important part is that the oxygen probe is very accurate, the chamber is very precise, and we managed to keep the water at 35.5 degrees consistent without any problem. And that's like a closed calorimeter at the end, and you throw a chunk of liver there. That only thing that happens is if the tissue is alive, is to consume oxygen. So that's how we get this curve here. We have a decay on oxygen, and then based on the amount of sample that you put and the decay of that, you can calculate the oxygen consumption of the liver sample that you put in there per grams, basically. And it was worth all the work on this. And this is what we observe here. We observe that our efficient heifers, they not only have a higher oxygen consumption for the whole body, but also higher oxygen consumption in the liver tissue, which is about 65% higher than what we have for the inefficient ones. And interestingly, this oxygen consumption by the liver was also correlated to T3 and T4 of about 40%. So it's kind of interesting and nice to see that things are coming along very well in here. And we are managed to get some useful information and in a very comprehensive manner. Another work that we did with those heifers at the same time that we did calorimetry was also to do infrared thermography. So we took pictures of 7 different locations across the animal at 1 hour to do this infrared imaging work, and we found some interesting relationships in here as well. So this is like the 2 body locations that matters most. So on top, that's what we call is node, which is just like that little curve. The drums are the areas that we target for temperature. So that's just just around the nose there, like it's extremely the body with a lot of capillarity, so it's very important for heat dissipation. And at the bottom here, the foot of the animal, which is also quite important for heat dissipation. And we found there a negative association with T4. Okay, well, how to explain that? So I was reading a lot these days about the fate of oxygen, and I found a paper from Leaks on 1945 that he talks about the fate of oxygen, because we always think of like oxygen combustion and heat production, but in fact oxygen can have quite a few other fates in our body, and sometimes people just forget about that. So what you're suggesting here with this correlation is that animals that are more efficient, they are better in conserving heat. And that really goes back to 1940, 1950, when we talk about Kleber, Blackster, and Brody, where they always make the analogy of animal as a machine, and a better machine is the one that can conserve more heat. So I think that what they're seeing for these animals here make a lot of sense. And also I found very important to acknowledge this paper from Whittle, 1962, because that was one of the it was a very important paper when I found this, because I was there like working with infrared and think that I was doing the most novel thing. But then Whittle already had to figure out that how important are the extremities for thermoregulation. So we're just using advanced equipment to do the similar thing in a different application. Okay, so that's all for the heifers. They all calved healthily, 36 beautiful calves. No issues here. And then we'll be change our gears a bit. We'll be talking now about bulls. Feedlot bulls, which is pretty common in Canada to feedlot animals, bulls, and then from there we have a bull sale or they're sent to slaughter. So just a little bit of recap here. So far I show you some relationships between T3, T4, and whole body, or to some extent liver, which can be much more than that, but a little bit of liver. So now we'll be moving that to a higher level. We'll also be talking about pituitary gland and how that affects the thyroid through TSH, and also the relationships of T3 and T4 with some other systems. So basically male reproduction and heart. I started the heart. So last year we are very happy that one of our lab members was awarded Best Award Representation from the EAAP in Warsaw. Jasper, on that talk, he's talked about heart physiology and structure as it relates to feed efficiency. So I want to build up a little bit on that. This is pretty much the bottom line of his work. So basically here in this slide, I'm picturing to you what's the heart of an inefficient animal. So when it comes to structure, an inefficient heart has a lower right ventricle weight, has a thinner right ventricle wall, and also has a larger myocyte width, so when you do histology. And when it comes to function, an inefficient heart has increased resting heart rate, has diminished stroke volume, and also larger blood volume. So some of these features are related to T3, T4, and TSH, and I will present that to you right now. So let's start with some of the structure and blood volume here. We found a strong association between blood volume and T3, which is quite interesting. It seems that you have more metabolism going on, you probably need more blood circulating. And then when it comes to structure, we found positive correlations between T3 and myocyte width and circumference length. So this is basically telling us that the more T3, you'll be pushing your heart to a higher level of capacity to work. You'll be stretching the fibers, you'll be making that heart, that muscle to work more than would happen if you have less T3. And another thing that's quite interesting, we've got correlations of the similar magnitude, but in the opposite direction when looked at TSH. And that also took me a little bit of time to wrap up my mind around that. But basically, TSH is an interesting hormone. To make that short, if you have a lot of TSH in your blood, that means that your thyroid is not working that much. So it's always an inverse relationship. So the fact that you have low negative relationship with TSH and a positive with T3 makes complete sense in this context here. And I can expand that with, to you later on if it's needed. Okay, so now we'll be checking what happens to those correlations when you look at the animal during resting. So this is basically this drawing is just a paper that was published these days on heart rate overnight, and I just took advantage to put in here. And basically shows you that overnight, inefficient heart has a higher heartbeat than the efficient one consistently. And we like to work a lot with overnight because that's where you can capture most of the resting metabolism, or like when the animals are actually laying down, there's not much going on the farm. And we find some interesting correlations here. Again, TSH and T3, they have some correlations, with T3 having a positive correlation of 0.55. No correlation with T4. I wanted to really pay attention to these correlations here to what happens in the resting metabolism or the overnight, because now we'll be moving that with the animals in different scenarios. we'll be checking the same animals, but when they are like in transportation, when they're taking them from the farm to slaughter, and when they're taking the last step towards the stunning box. And what happens with TSH? TSH was negatively associated with heart rate during the transportation, no association whatsoever with T3, and then a very strong correlation with T4 and the heartbeats in the very last few minutes of the animal's life. So that kind of makes sense to me, because the animal was like in homeostasis, and then you push that animal to a truck, that breaks the homeostasis. So one of the first things to do, okay, you need TSH, you need to be ready for quite a few things here. And after that, which like between transportation and stand is about 1 hour, a lot of TSH really cause a lot of T4 to be released in the bloodstream. So it's quite interesting to see how all that biology happening while they're just moving animals from one place to another. We also caught some correlations here, because I talk about heart. It's also important to talk a little bit about blood. So there's moderate relationships between T3 and T4 with both red and white blood cells. There is more to learn about that. This is like a very basic assessment of a CBC, so there's quite a bit more work to be done on that. Okay, so now for the last few minutes here, I'll be talking a lot about how T3 and T4 can be related to reproduction. And it's funny because it was never my intention to work with reproduction. It just happens because I was willing to help a friend, and then my friend just gave up an experiment, and then okay, I can take it over, and then one experiment leads to another, and then we end up getting a lot of stuff on the both sides. And one particular type of bull is the one that I have been working with. That's the young bull, which is a very tricky animal. That's the animal that has about 12 to 14 months. That's not, it's still in development. There's quite a bit of variation, looks at different breeds. So it's not an easy material to work with. And this is a summary of some of the evidences that we have been observing for the relationship of feed efficiency and sexual development bulls. I started this with one or two assessments, but then I realized that having a lot of students and doing that around the shoot was too much cha-cha-cha talking. So then I decided to put a few more assessments. So then is everybody pretty busy? Then the thing just move faster then. And so these are pretty much the assessments that they're doing. On top we have a scrotal circumference. Of course, that the other one is histology, that we do that with abattoir pieces. And then this gray, black and white one here is a pixel intensity, which is a very interesting assessment, that we'll be talking more about that. And then we have morphology of the semen, and also the infrared imaging, or the infrared spectrum of the scrot of the bull. And based on all these different assessments, what we're observing is that the inefficient bull, he's better when it comes to sexual development. He's a few steps forward than the inefficient bulls, which might not be a big deal for many people, but when it comes to the industry, that can be like a major thing. So we have been working around this a bit. Okay, so pixel intensity. This one is quite interesting. It takes 1 minute to do that ultrasound, and then we find a positive correlation with T3 and T4 and with the TSH. So basically here, the higher the pixel intensity, the more structure you have in the testicle, the more developed it is the testicle. So it is association with development. Another thing that we did was to check their associations with different types of testicular tubuli. So we have immature, 3 classes: immature, reach maturity, and mature. And then we check some of the correlations on that. So the proportion of immature was positively correlated to T4, while when you look to the areas of the tubules, either at the testes and also at the epididymis, we found a negative association with T4. So here is it's a bit hard to understand that, because it seems that if you are in a higher level of development, you have different amounts of T4. We still have more work to do on this. I just want to share it to you that we have observed some associations there, but there is more to understand here. And the same is true when it comes to semen quality, and it's even trickier, because semen quality, the sample that we got today has a history of about 70 days for when that semen was first produced. First initiated, and what you have observed here is a positive association. So meaning that you have more defects if you have higher levels of T3 and T4. So that kind of goes in an opposite direction to what we found in pixel intensity. But again, spermatogenesis is a process that takes 70 days, and what you're doing here is dealing with younger bulls and working with their first ejaculated. So there's quite a few limitations here that you need to work around. Okay, so we kill those animals, and then we get quite a few samples from inside of them. Here what matters for now is the pituitary gland, that's that little one there, and we talk a little bit about that. So this is a H&E image of the pituitary gland. We have quite a few different pieces of the pituitary gland, different regions, and what we're really more concerned with here is the anterior and posterior pituitary. And we find some interesting correlations there. This is the proportion of the whole gland that's taken by either anterior pituitary or posterior pituitary. So when it comes to anterior pituitary, the inefficient bulls, they had larger area in that region, which again makes sense because anterior pituitary is where most of the thyrotropins are produced, so they have a hard structure to the developing hormones that might be affecting thyroid. And also the posterior pituitary, that you had some difference in there. Okay, before they shoot me in the stage here, just go quickly to some cell metrics here. So we measure the lumen of the thyroid gland, the area of the lumen, and what's taken by the luminal space and what's taken by the epithelium. And also did quite a few measurements in the thyroid cells. And this is some correlations that we observed. We observed a positive correlation with T4 and follicle size, which made complete sense, as we know that those follicles are really like little swimming pools of T4. Also we found some interesting associations when it comes to cell area and nuclear area with TSH and T4. And also some difference here when it comes to the metrics that are more important for now. So it seems that our efficient animals have larger follicles, and then when it comes to cell to nuclear ratio, the inefficient animals, they have a higher cell to nuclear ratio, which is really an indication of a higher metabolism on that cell producing hormone when you have more cytocell and smaller nuclei. We had some similar work that we did in the intestine along the same lines of histomorphometry. And the intestine, just to give you an idea of another example, we observed that the number of cells in the duodenum and ileum were increased in the efficient animal in comparison to inefficient animal. So really suggesting that those animals have a larger cellularity and a higher capacity to absorb nutrients. If you want to see a bit more about histomorphometry, in this case applied to rumen, I invite you to see section 16, the presentation by Len, where I should be showing quite a few interesting associations with feed efficiency and rumen structure. So just final remarks here. With this presentation, you could see that there's quite a few ties between productive performance, reproduction, welfare, and health that I relate to the thyroid gland. And it's kind of nice when you can blend that with the physiological aspect of husbandry, and from there really deliver what you're really after at the end of the day, that there are refined phenotypes that could be used to support other disciplines, and from there leading to develop of multi-stream and multidisciplinary technologies. I think that this integrated view of physiology and other disciplines is quite important, it's quite decisive, and it's really missing. With this, I just would like to acknowledge a lot of in-kind and cash support that has been supporting my research program, and I can take some questions that you may have right now.
There's one question popping up.
Thank you for a very interesting and stimulating presentation. I'd be interested to know how you reconcile your observations in the pregnant heifers with the laws of thermodynamics. Because the only way an animal can grow, and the fetus can grow, is through the free energy, which is determined by the balance between the enthalpy content and the entropy change in a system. As the entropy change is the extra heat, and you've got animals that are not losing massive amounts of weight, how do you reconcile your observations on heat production with those laws of thermodynamics? Because there is no, thermodynamically, there's no such thing as a free lunch.
Well, I think that the first part, that might be a limitation of our experiment. We're really there like measuring oxygen consumption, not heat production. Although those things are quite associated, they're not like not exactly the same thing. So that can be something to be considered. And I think that's probably my main consideration about that. And certainly, like, this is one experiment. There's much more to be done on that, and I hope to be working on that further in my career. But I don't have much more explanation on that. I need to chew more on that and certainly learn a bit more.
Just as an addendum to that,
Yeah.
if you measure the body temperature,
Yeah.
and you've got the surface temperatures that you can readily take from thermography,
Yeah.
then if you calculate the average, the gradient, you can actually work out the heat content of the animal, which
Okay.
should change
Mm-hmm.
if that's one of the components of the equation. The heat storage should go up.
Yeah.
And you can actually calculate that in terms of energy rather than just change your body temperature.
Yeah, and that's probably a good thing to do, because you have the thermal image across different locations of the body, so we can do a complex work on that. That's great. Thank you.
There's a further question?
Again, great talk. I just wonder if I could make a suggestion as an overall summary of what you've presented. Is the major mechanism of the greater efficiency of your animals simply a reduction in basal metabolic rate, and therefore maintenance requirement for energy? So that when you compare them statistically or mathematically at the same feed intake, you've simply got more energy available for growth.
I think that if you need to put that in a sentence, that's what it is. But I really need to see that that change in metabolic rate is really like a lot of shift that's happened there. Because we have like a lot of evidence that like animals that are more efficient, they have like a more cellularity in the intestine. They have a higher metabolic rate of the liver. And in the classical side of it, when you look to energy metabolism, you want to decrease the maintenance of those organs, so then you can have more production. But in many instances, you're seeing that works a bit different. You have like a more functional intestine with more cells. We have larger cells in the liver, and that's kind of not quite always the case when you look to classical energy metabolism.
There are more cells in the body than the intestinal cells.
Yeah. Yes, exactly. Yeah, and when you look to immune system, it's also interesting because I didn't present here today, but animals that are more efficient, they seem to have a better immune response. And again, we always blame immune response as a kind of waste of energy, which I don't think that's the case. So.
Maybe look at the energy cost of the muscle mass in the animal.
Yeah.
That might be the main driver.
Well, thank you again.